Okay, we're coming into the home stretch of this particular PowerPoint. So, ooh, I want to go back to, sorry, the cytoskeleton. Sorry about that. Okay, so starting up on the cytoskeleton, so we're going to kind of highlight a couple of the organelles specifically, especially the ones that weren't talked about in great detail in the previous part. So the cytoskeleton, super dynamic. Dynamic just means that it can change and shift and move. And so the function is going to be based on what does the cell need at that particular time. Sometimes it is shape and internal framework. Sometimes it's strength, integrity of the actual cell, anchoring, changes in the shape or position of the nuclear membranes and organelles. It allows movement, so it can actually move things back and forth across the cell. So when we were talking about vesicles moving across that cell to get to the plasma membrane, that's being allowed by the cytoskeleton. It can also perform a handful of different cell type functions. Phagocytosis, so it can enclose things. It can contract if it's a muscle cell. So cytoskeleton does do quite a bit of functions for us. So there are basically three main components of any good cytoskeleton. They are protein based. We had talked briefly about when proteins are used structurally. Most of the time, they are in fibers, and so we call them filaments. So we have actin filaments, we have intermediate filaments, and then we have microtubules. An actin filament can also be called a microfilament, so sometimes the text will use that term. It's the thinnest of the filaments. It's composed of two intertwining strands of actin subunits. Support, it bears tension, it can help maintain the cell shape, and it's also involved in moving the motor units, which is going to allow these little, they look like little olives, but these little pits in the actin is able to interact with myosin. Intermediate filaments are more rope-like. They are a different type of filaments, fibrous protein. They're built from keratin, so they're much stronger. They're also much more permanent. There's less shifting and moving of these. These tend to just stay for framework and anchoring. They help maintain our size, and they also help us withstand some of the mechanical stress that is applied to a cell. Microtubules, they're the largest of the filaments. They are hollow, so you can kind of see how it's going to create a hollow tube once they're arranged. And it's created through tubulin, which is, again, a different type of protein. It can be rapidly added or removed, which means it can alter the shape of the cell fairly quickly if that's a need for that cell. It also plays a role in the motor potential and transporting things around. So when I talked about the vesicles being allowed to move, that's primarily through microtubule movement. Here's a nice table for those. They show the illustrated version, and then they have a light microscope. These are light microscopes. You see the LM light microscopes, and these are fluorescently dyed so that you can see the difference in kind of their composition. Okay, and it also has their function there. Types of filaments, it's you can make all kinds of different things from these. So you can have centrosomes. That's where you had the centrioles. This is going to allow for anchoring during cell division. So that's what those look like. You can see it's a bundle. It's a little bundle of these microtubules. So if I cut it, a transverse cut across that, it's very pretty. And this is a transmission electron microscope, but you can see the hollow tubes of the microtubules there. Some other things that can form from the cytoskeleton: microvilli, cilia, and flagella. So a microvilli is like what we see on the surface of our small intestine. It will increase surface area, which for our small intestine is where we absorb all of our nutrients. So having increased surface area, because that's what folded membranes basically do, having these increased surface areas allow us to absorb more. Now, microvilli and cilia are very similar. The biggest difference is these are not going to be used for movement. These are primarily going to be used for surface area and absorption. So when you look at cilia, they are also hair-like projections, also made of microtubules and some motor proteins. But the thing here is cilia has the ability to shift and move. So it's either moving things across the surface, or it's like the cilia that you have in your ear, for example, that helps with balance and hearing. The flagella is a solitary long structure. You'll have a lot less of these. The only one that's found in the human body is actually sperm, and it does have a whip-like fashion to propel it forward. Both flagella and cilia are very similar to a centriole. The biggest difference is there's not a central set of tubes. And so it has a slightly different shape, but it's still built similar. So you can see that design looks very similar to the cross section that we saw of the centrioles. Okay, again, a nice little table for that. Looking at the nucleus, this of course is the control center, but what it's controlling is primarily cellular component creation. So it does house the DNA. This is the codes for pretty much everything that occurs in a cell and in your body. The plans, the manual. Genes are going to be segments of DNA that are coded for a particular trait or a particular hormone or a particular protein. And we will have to take DNA and turn it into RNA. Within the nuclear envelope, this is again the membrane surrounding the nucleus. It will have pores, but inside of the nucleus it has its own concentration of plasm. So it's called nucleoplasm. It does contain cytosol. It's very similar to cytoplasm, but it's going to have a different composition as far as what's dissolved in it. So it does get a different name because, of course, you have nucleotides in there, you have enzymes, you have different proteins, DNA, RNA, that you won't find just in cytoplasm. And again, if you see the term chromatin, this is implying loose DNA. We will also have another structure found within the nucleus called the nucleolus. And the nucleolus is going to be suspended in that nucleoplasm, and it's going to create ribosomes for us. So here's that image. You can see through the electron microscope the openings, the pores here on that surface. So the nuclear envelope is also a double phospholipid bilayer. So just like the mitochondria, there's two layers surrounding it. There's an outermost membrane that can have ribosomes, and it also continuously touches portions of the endoplasmic reticulum. There's an inner membrane that is going to be supported by intermediate filaments. And then, of course, your pores are going to be openings that allow movement between the two parts, the interior and the exterior. So there's another image of that pore. And certain things like RNA, for example, are small enough to exit through those pores. Chromatin, which is DNA, loose DNA, has to be folded and shifted for space reasons. So what happens is there's these little proteins called histones that kind of act like the reel on a rod and reel. And they will actually twist the DNA up. And once you have a histone wrapped around, or some DNA wrapped around a histone, that's called a nucleosome. And what we can do is we actually can tightly condense this DNA material. So when we need to divide our cells, we do actually condense them down, which means that those histones will start to coil up and wrap more DNA, and then the histones will start to incorporate and interact with each other. And so we can really tightly pack our DNA into about 46 chromosomes. So most of you have heard of a chromosome. This is just tightly packed DNA using histones and what's called supercoiling. On each chromosome, you will have what's called a sister chromatid. This is identical copies. So if I'm making a new cell, I need to make sure that what my new cell has the same stuff as my old cell. So I will make identical copies, and they will be connected at what's called the centromere. So here's this process. We have DNA, and that DNA will start wrapping around histones, forming the nucleosome. Those histones will start to, you can see they're starting to interact with each other and do what's called supercoiling. That supercoiling will then form our iconic X shape. The sister chromatids of this side is a sister to this side, so they're identical and connected at the centromere. So this is how we end up seeing the appearance of our chromosomes. It's called a karyotype, which is a illustrated view of our chromosomes. Okay. The nucleolus, or nucleoli is plural, but singular is nucleolus, is going to be responsible for creating ribosomes. So it will have what's called ribosomal RNA in order to create those ribosomes. I will stop here for this